The next talk is from Jack Gallant, and the title of his talk is Modeling and Decoding the Human Brain, so Illa's talk was a great lead-in. Jack is at UC Berkeley, and he's another one of these labs that really takes a diversity of approaches from classical electrophysiology to human brain imaging to computational theoretical approaches to tackle sort of what does the visual system do, and he's going to talk to us about decoding.
So the title of my talk was Modeling and Decoding the Human Brain, but really I'm going to talk about the search for functional maps. That's OK. It's pretty close. They're all related. Encoding models, decoding models, and functional maps are all closely related. Now, over the last 20 years, there's been a remarkable explosion in technologies that allow us to measure the brain. Of course, we notice this mostly in molecular biology, where there's a new technique every week. But even in studies of big brains, like human brains, there's been some remarkable developments that have allowed us to do things that we couldn't even think about doing 20 years ago. And the most remarkable method that's been developed during this time is functional MRI. Now, functional MRI doesn't measure neurons. It measures blood flow, changes in blood flow, blood oxygenation, and blood volume that are consequent to neural activity. But because MRI is a three-dimensional technology, it provides better signals about the human brain and what's happening there than any other method we have available today. So over the last 20 years, functional MRI labs have spent a lot of time working on sort of the classical approach to studying the human brain, which searches for functional localization. This is an approach that dates back to the early neurology studies back in the 18th century. And today, we still see it alive and well in functional MRI. And this has been a pretty successful approach thus far. These are the areas painted on the inflated brain. You can see that there's a lot of functionally identified areas that are known in general. It's known in general what their function is. The visual system's back here in the back where it should be. We've got the somatosensory and motor systems. We've got some speech areas that have been identified. And this is all great. It's worked really well. But my view is that just doing functional localization is necessary but not sufficient for saying we understand the brain. And the reason is my definition of understanding is that you have to be able to build a predictive model. You have to be able to build a model of the system you're trying to understand that faced with an entirely novel situation that you have no idea how the model should be relevant, that you will actually accurately predict brain activity in those situations. And mere functional localization doesn't give you that. You need to do something more. So I tend to think of this like a reverse engineering problem. So on the left, we have our brain. It's got a bunch of mysterious parts. And on the right, we have Intel CPU chip. It's got a bunch of mysterious parts. And you can imagine that trying to reverse engineer this chip would be as difficult as trying to reverse engineer the brain. But in a lot of ways, the problems are quite similar. This chip has a bunch of different parts. And if you just did functional localization, you might say, well, OK, I think this is probably one part of this circuit, and this is probably one part. But it wouldn't really tell you exactly what the individual parts do. And especially in the case of a computer, you wouldn't really have an idea of how it worked if all you knew was what the parts were, because, of course, you can run almost any program on this chip. Now, in the brain, the problem is in some ways more difficult and in some ways less difficult. It's not a design system. It's an evolved system, so it's fairly complicated. But of course, on a short time scale, you can't change the connections in the brain. So the function and the connectivity are very closely intertwined. So you can imagine trying to understand the brain by either attacking connectivity, and I think David Metis is going to talk about that later today, or trying to attack function at a large scale. And that's kind of my approach. Now, if you're thinking about trying to map function across the brain, then the first thing you think about is maps. Not all quantities in the brain are represented in maps, but a lot of things are represented in maps. And it seems that when people look at a finer and finer scale and they get more and better data, they always end up finding, or often end up finding, that information is organized according to a map. So if you look at in area A1, you can find tonotopic maps. If you look in classic whisker barrel field and in rats and mice and other rodents, you find whisker maps. And you can use MRI. It's well known to discover maps in the human brain, too. The most successful mapping method in the human brain has been to find retinotopic maps of angle and eccentricity, and therefore to use those to delineate the retinotopically organized visual areas at early stages of visual processing. The question is, can we use this kind of vague, mushy MRI technique or technology to discover other kinds of maps in the human brain that would allow us to elucidate other kinds of functions that aren't just low-level retinotopic vision? So to think about how to do that, let's take a step back and just think about the kind of data we're going to collect with this technology, because fundamentally everything we do in MRI is limited by the quality and kind of the data we're getting. So on the left here, we've got a movie that somebody's watching while they're in the magnet. On the right, we've got a brain. Of course, the brain is inconveniently folded up and stuffed inside the skull. So if you come from the David Van Essen lab, you always flatten out the brain as the first thing you do, because it makes it easier to look at. And then here on the left, medial on the slide, we have the visual system. And here's the front of the brain, here's the top and the bottom. And you can see that as this movie's playing, we get a lot of changes in blood flow. This is a map, essentially, of blood flow. And blue means less than average blood flow, and red means more than average blood flow over time. You can see that blood flow is changing most in the back of the brain, in the visual system here, but it's also changing up here in the front, although to a lesser extent. And this makes sense because in this particular experiment, the subjects weren't doing anything. They were just staring at a dot in the middle of the display. They're not actually doing anything at all with the information. So if you want to understand how the brain encodes these movies, you can think of this as a simple multivariate regression problem. I have a bunch of information in this movie. I've got pixels, I've got people, I've got objects and actions, and then I've got a bunch of points on this map in the cortex. In a typical 3T MRI machine, we're going to have 30,000 points. And so I essentially have to build 30,000 models that allow me to predict from this movie what the brain activity is that will be elicited by this movie. And of course, if the brain actually has different areas, say there's 200 or 300 different brain areas, then what I'm really going to need is 200 or 300 different models, each one of which describes how the movie is represented in that particular location in the brain. And of course, I don't know what most of these brain areas are beforehand, so this is sort of an exploratory data analysis problem. I have to fish around to model the brain and try to come up with systematic relationships between the movie and the brain. Now, this seems like a weird thing to do, but actually, we've been doing this for a long time in neurophysiology, where we record from single neurons. And we may be recording from a neuron in a part of the brain we're not really sure what it does, and we're not really sure what the feature space is that it codes in. And so we developed a systematic processing pipeline that allows us to model neurons even if we don't know beforehand what they're supposed to be doing. And we can apply this same logic to MRI. So it essentially has 4 stages. We measure some brain activity. We collect a bunch of data sets, one to fit the model, one to validate the model. Then we estimate our models. And we're going to do this for each individual location in the brain. As I said, we're measuring 30,000 voxels on the cortex, so we're going to do this for 30,000 individual places in the brain. We specify a feature space. We fit the model, and then we evaluate predictions. And if this location of the brain gets good predictions, then we might say, OK, I'm done. If not, then we might go back and keep doing this over and over and over again. And in fact, we have dozens of models that we fit to the brain all the time. And then once you get predictions for a location of the brain that you're happy with, then you can try to figure out why your model is doing what it's doing. So you can visualize receptive fields. These could be in neurons or voxels. You can visualize the weights, the individual weights for the individual models in the feature space. And you can visualize them in the cortical surface. And this is where you're going to discover maps. And then at the end of all this is the science part. At the end of all this, you can do decoding. This isn't science at all. This is just engineering. You kind of get decoding for free in this framework. But the advantage to decoding is it's obvious what's going on if you look at a decoded result, and it's very compelling. So I'm going to show you just one example of the dozens of kinds of models we fit. I'm just going to show you one example that illustrates how we can use this kind of abstract modeling framework to recover maps for higher order visual information that you wouldn't necessarily think would be mapped across cortex. And in this case, we're interested in mapping, trying to see if there's a map for categories, namely object and action categories. So to do this, we first of all have to propose a feature space. And linguists have conveniently developed this thing called WordNet that's essentially is a hierarchy of all the nouns in the English language. So at the root of this whole thing is this thing here, which is thing. And then growing out from the thing branch is here's people and animals. Here's indoor scenes, outdoor scenes, vehicles, boats, and artifacts. And these are other strange things like body parts, atmospheric phenomena, textures, and so on, text. And then there are a bunch of other is-a trees for verbs, like the communication verbs, traveling verbs, and change verbs. So we essentially took the 2,000 most common nouns and verbs in the English language, and we said, OK, that's our feature space. We went through the movies, and we encoded all of the objects and actions in the movies, and then we used them to fit models for all the voxels in the brain. And after you do this, the very first thing you need to do is you need to check the predictions of your models, because you don't want to be trying to make decisions about a model that predicts poorly. That's the wrong model. So here we have the inflated brain and a flattened brain. And the color map here simply shows the quality of the predictions of this model at each location in the brain. This is the retinotopic visual areas here at the back, and this bright yellow sort of semicircle, well, it's dorsal and ventral here, is the higher order visual areas that sort of surround the retinotopic visual areas at the back of the brain. You can see that this model actually fits pretty well in these higher order visual areas, and I've noted in here with white lines some of the functionally localized areas that some of you may have heard about before, such as parahippocampal place area, the fusiform face area, the exostriate body area, and other kinds of functionally localized areas. So this model seems like it works pretty well in these areas. That means we can go ahead and actually try to figure out what the model's doing. So the first thing we can do is look at the receptive field for a single location in the brain. This happens to be one voxel in the right fusiform face area. And on this plot, I've shown you, again, the WordNet graph. Red here means nouns and verbs that elicit more blood flow than average, and blue means nouns and verbs that elicit less blood flow than average, and the size of the ball here is just the magnitude of the effect. And you can see that this particular voxel that's in this area known as fusiform face area, it responds pretty much to any kind of person or athlete or people or men and women, whatever. Also any kind of animal with a face like a carnivore, an ungulate, also to all kinds of communication verbs, and then to some other weird things like communication, some strange attributes and legs. So, okay, you could do this for all 30,000 voxels in the brain. That would take forever. It would drive all of us crazy. So you need to do dimensionality reduction somehow or other. This is essentially a data matrix where we have 2,000 semantic categories and 30,000 voxels. And if you're stuck with this kind of data, the very first thing you usually do in a data analysis method is do principal components analysis. When you do principal components analysis, to make a long story short, across human subjects, you find out that all humans seem to share a common low-dimensional 4 or 5-dimensional space that organizes these semantic concepts in all human brains. Now, humans probably share more than 4 or 5 dimensions, but because of the limitations on simultaneous noise and MRI, we can only see 4 or 5 of those dimensions in common across people. And now that we know that humans have 4 or 5 dimensions in common, we can visualize them. And the first thing we can do is we can take the first 4 principal components and project them into the feature space. That's shown here. So this is the first principal component up here on the left. And the first principal component in this kind of data analysis problem is always essentially energy or variance. And here we have essentially bright, fast-moving things versus slow, dark things. And that kind of makes sense because bright, fast-moving things are going to elicit more neural activity pretty much everywhere in the visual system. They're going to elicit more blood flow. Now we can take the next 3 principal components, 2 through 4, and we can project them into the feature space using a color code where the RGB coordinates are labeled according to the color of the projection into the feature space. And you can see here the results for the other 3 principal components. And essentially, neighboring colors in this space indicate concepts that are represented similarly in the brain. So the brain represents all kinds of animals similarly, all kinds of people similarly, although athletes are a little different. It represents vehicles, all this kind of the same. Represents all kinds of indoor scenes, buildings, shops, and rooms the same. There's a lot of information here. You can kind of go through this map fairly slowly, a piece at a time, trying to pull out what the individual concepts are that are sort of form microclusters inside this large space. But that's not the goal of today. The goal of today is actually to think about, OK, how is this information represented on the surface of cortex? And to do that, we can simply take this giant piece of data and project it onto the cortical flat maps using the same color scheme that we have here. And that's shown here. So here we have the right and left hemispheres. I've put only 16 of the 2,000, roughly, categories up here as a color map, just to give you an idea for what's happening. Remember, green here means people and body parts. Purple means vehicles. And yellow means kind of animals. And you can see that pretty much all of exostriate visual cortex, beyond the retinotopic areas, the higher order visual cortex, is covered with these large gradients of semantic selectivity for both nouns and verbs. And these aren't discontinuous. They're not little punctate peaks of activity. They're not individual, localized, highly segregated functional areas. It's just a continuous map of gradients of different kinds of semantic selectivity. And the way I tend to think of this is that really you have a 4-dimensional space, these principal components, and then you have to project this 4-dimensional space onto a 2-dimensional cortical sheet. And so you end up with a slightly fractured map because the projection from 4 dimensions down to 2 dimensions can't be done perfectly. And then the visual areas that we all know about, like the exostriate body area, the fusiform face area, all these classic functionally localized areas, these are essentially the nodal points on this map. They're the most homogeneous regions on the map where all of the voxels in that local area tend to be similarly tuned. But really, the visual information is tiling this entire map and covering it completely. Now, it turns out, as I mentioned earlier, that once you have an encoding model, you get decoding for free. So an encoding model basically takes a stimulus and nominally maps it into some feature space where you can linearly map between that feature space and the measures that you make, bold, in this case, blood oxygen level dependent responses. And once you have this, you can nonlinearly map by taking a bunch of bold activity that you measured and mapping it back into the feature space and then nonlinearly mapping it back into the stimulus and reconstruct the stimulus, if you so choose. And this is essentially just solved by Bayes' theorem. So again, once you have an encoding model, you get decoding for free. The better your encoding model is, the better predictions it gives you, the better your decoding will be. And anything you can build an encoding model for, you can immediately do brain decoding. So this is brain decoding from the WordNet model. We're decoding 2,000 semantic concepts here, 2,000 nouns and verbs simultaneously from one person's brain. On the left are movies. These movies are novel and were not used to build the model at all. Person was fixating in the middle here. On the right are the concepts, and they're just mapped in two principal components so that they don't overlie each other. And you can see that this thing works pretty well. Here's a dog there, there's a man, they're talking, they're in a room, there's a face. Now this part it has a little trouble with. It knows there's a street and road, but it's very fast. Okay, so now we switch to an ocean scene. We get sky, clouds. Now we're in Waikiki, so we get buildings, body of water, mountain roads, people walking, sky, trees. That seems to work pretty well. Now we switch to some women. First of all, it says, oh, there's some text. You'll notice sometimes the words appear a bit before the movie. That's not because the person has ESP. It's just that we don't have the timing quite right on our decoder. Here's a woman talking, face. Here we go back to an ocean scene. We get fish, swimming, water, shark. There's no shark. But OK, this is actually a manatee. It thinks it's a whale because manatee isn't in our data set. But that's pretty good. Guessing a whale for a manatee is pretty close. So you can see that this thing works amazingly well, right? In fact, this actually works well enough where it kind of scares me that it works so well. We're decoding a lot of information simultaneously using MRI, which is frankly a pretty primitive technology. I mean, it's an advanced technology, but relative to what we would like to have, it's primitive. It's measuring a small echo of what the information is actually that's in the brain. Okay. Now, at this point, oftentimes in a talk, I go through all the different kinds of decoding models we have because people really love decoding models because they're kind of compelling. But today, I thought I would change it up a little bit and talk about something new, which is what happens to these maps under attention, right? The connections in your brain are fixed on a short time scale, but obviously you can attend to different things as you need to in order to perform a task and get around the world. And the question is, what happens to these maps when you attend? Now, there's a lot of neurophysiology that suggests that as you go more and more anterior in the brain, you end up with these maps, or the tuning of individual neurons, becoming more and more flexible and changing more over time. Earl Miller's got some great data on this from the frontal cortex. My lab has shown that you end up with tuning curve changes under different states of attention, even as early as area V4. So we decided to set out to see if we could look for tuning changes in these maps. And the reason is we realized that tuning changes in individual neurons will lead to warping of the semantic space, the way that the categories are represented in the brain. So if you think of a space of semantic categories, these live in our data in some four-dimensional space. And you can imagine that when you attend to humans, what might happen is that humans become represented sort of preferentially, and that the representation of all other things that are non-humans kind of get crushed together. And then if you attend to another thing like vehicles, then vehicles get represented preferentially, and all the other concepts get crushed together. And that would happen if you have neurons, or voxels, that tile the space, and then when you attend to one concept, say humans, the voxels tend to shift their receptive fields over towards humans. When you attend to vehicles, they shift their receptive field over to vehicles. This is, in engineering, is called a matched filter. You're essentially building an optimal filter by changing the tuning of all the individual constituent filters to represent the part of visual space that's behaviorally relevant in a much higher resolution. So to do this, we did a really simple experiment. We showed people movies again, and we just told them, in one condition, search for humans. And every time you see humans, press a button. In another condition, search for vehicles. Every time you see vehicles, press a button. It's a really easy experiment. Subjects like it. You can look at single receptive fields. So this is actually another voxel in the fusiform face area, FFA. And when you search for humans, it looks like a face voxel. It's tuned to people and animals with faces. But then when you search for vehicles, although this voxel maintains its normal tuning, it also now becomes tuned for vehicles and other kinds of artifacts, which it wasn't tuned for before. So this shows that on a single cell, excuse me, these are not neurons. They're not even close to neurons. They're voxels. On a single voxel level, this voxel's shifting its tuning from one sort of place in this semantic space to another place in the semantic space, although it's not really shifting. What it's more doing is sort of lifting up a leg and allowing some information to pass that it didn't pass before. Now, if you do this for all the voxels in the brain, you end up with a map like this. On the top is a map of all the voxels. The red are the voxels that tend to shift toward the target. The blue are the voxels that tend to shift away from the target. This is for humans, and this is for vehicles. And you can see that a large fraction of the voxels tend to shift towards humans or towards vehicles as you would tend to humans or vehicles. So its tuning curve shifts are quite dramatic. And here they are mapped across the surface of cortex. So these tuning changes are widespread. They appear almost everywhere in the brain. Interestingly, there are a few places where you actually see shifts away. These end up being certain locations in frontal cortex, the temporoparietal junction, and the precuneus. And those are actually three constituent locations in part of what is known as the default network. Now, the map I just showed you, you might say, well, wait, there's an artifact here, right? Say you're doing this task, and I tell you to search for humans. And so all of a sudden, you see a human. You get excited. I see a human. I see a human. You hit the button. You're all happy. You're excited. Your blood pressure's gone up. Your blood pressure's gone up. Your bold signals go up in your brain. And this is an all artifact of target detection. So to get around that, what we did is we went through the data, and we removed all of the situations where humans or vehicles occurred, and we refit all the models. Now, we can't look at the tuning for humans or vehicles because they no longer appear in the movies, but we can look to see if all of the other constituent categories shift towards humans or vehicles when you're searching for those categories, and indeed they do. Here's the baseline map, and you can see that when you're searching for humans, the map becomes much more yellow-green because now a lot of the voxels are shifting towards humans. And when you search for vehicles, the map becomes much more purple-pink because now all the voxels everywhere are sort of searching for vehicles. So what ends up happening is the semantic space warps around the concept that you're searching for because voxels all over the brain are essentially becoming optimized to search for this unique this particular category. So now I'd like to say what's wrong with this approach and why we're limited. And I've got just two things to say. First of all, this is a global mapping problem. We can't really recover local maps. So I showed you these retinotopic maps before. But if we look at just one location in the retinotopic map, this is the receptive field of a single voxel that should be tuned for orientation because we're recording here in area V1, primary visual cortex. If we're recording from a single neuron, it would look like a simple cell or a complex cell. But because we're recording from millions of neurons, you can see that the orientation tuning in this voxel is almost absent. And in fact, MRI, because it's measuring blood flow, is really measuring information that's globally mapped across an area, not information that's locally mapped in an area, although there is a small amount of local orientation bias as you go across the surface of V1. It's vanishingly small relative to the global variable that is correlated with the cortical magnification factor. So this is a serious problem. People are trying to address it by going to high field strength. If we go to 7T MRI, in theory, we should be able to do 4 times as good. In practice, it ends up being about twice as good, but that's better than nothing. So people are trying to do 7T MRI to do functional mapping. It turns out to be difficult, however, because of the motion of the brain due to the heart rate and breathing. The second big problem is, of course, that we're measuring the wrong thing, right? What we're measuring at 3T is big blood vessels. Most of the signals coming from blood vessels that are about the size of a voxel. That means blood vessels about 2 millimeters across. If we go to 7T, we can measure the capillary bed. But both of these things are the wrong thing because who really cares about blood flow, right? Only a neurologist cares about blood flow. No neuroscientist cares about blood flow. I don't care about blood flow. It's just a giant annoyance because if you build a model, you have all your latent variable neural model, then you have to have this ugly hemodynamic coupling term stuck at the end to model the relationship between neurons and blood flow. No one cares about blood flow. So we would like to get rid of this and just not be recording blood flow. People are trying to do that in MRI, but it's very, very hard because of the signal-to-noise limitations. However, even though we're essentially measuring the wrong thing and we're measuring it at low resolution, in fact, you can do a lot of things beyond the visual system. Just to give you a taste, this is the functional maps we get from essentially playing the same game we just played, but now for language. So we put people on the magnet, we play them stories. We generate a feature space that describes language. We take the principal components of that feature space and we map them onto the surface of cortex. And you get something like this. You can see that language is distributed all over the brain, a lot near the auditory system, but up here around the speech centers as well. You can see that there's hemispheric specialization between the left and right hemispheres, just as you'd expect because it's language. But this language map is quite rich and complicated. And in fact, you can decode language just about as well as you can decode vision. And that actually, I'll just end by pointing out that decoding language is very interesting future application, because if you can make a portable brain decoding device and you can decode internal speech, you now have essentially the most general brain decoding device you can possibly build because it's based on speech. And I think it's not going to be very long before we can decode the little man or woman in your head that talks to you all the time. And then it's just a matter of building a portable device, and that's just money, really. All right, so I'd like to just point out the three people who did most of the work: Shinji Nishimoto, Tolga Kucher, postdocs of mine, and Alex Huth, a great graduate student of mine, and my lab. Thanks very much for your time.